Quantitative PCR or qPCR is much like standard PCR, which we've talked about before. It's got many of the same elements: DNA polymerase, DNA template. You get your two primers, forward and reverse, your deoxynucleotide triphosphates, which are your building blocks, and then other ingredients just to keep the reaction going forward. What's unique about quantitative PCR is that you are measuring the accumulation of your product after each cycle or even continuously. And usually this works because you are measuring the increasing fluorescence of a reporter, which has been put into your mixture to tell you that DNA is being built. So it's kind of monitoring the activity of DNA polymerase. Quantitative PCR machines have these additional lasers or a single laser that moves around, and for each reaction that's happening, measures this fluorescence in real time. The output of this reaction is usually a graph where there's a y-axis and an x-axis. The x-axis represents time, oftentimes measured in cycle number. The y-axis measures the change in fluorescence over time. So the relative fluorescence of the fluorescent reporter is telling us essentially how much of my PCR product is accumulating. Now let's take, for example, gene A here in blue. It starts out relatively low fluorescence and then over time reaches a growth phase where the fluorescence rises almost in a linear fashion, and then will level off kind of like this, creating a curve. Now let's think for a moment about how this curve is being generated. We're using a forward and reverse primer that's targeting a particular gene, and the template that we're working off of in this example is actually complementary DNA, meaning DNA that represents all of the messenger RNA from a particular set of cells or tissue. Now for comparison, let's watch the amplification of a second gene, gene B, using a different set of primers in a different PCR reaction, but using the same template liver cell cDNA, which represents all of the mRNAs in that same sample. So you can see that the red line here doesn't accumulate until a little bit later in the cycle count and tops out at about the same level. Now, if we look at each of these lines, half max point, I'm just going to call it right here, and I draw a little dashed line down to the x-axis on each of them. We call this point where this line intersects the x-axis the CT value. We call that the cycle at which threshold was met. The threshold in this case is some arbitrary threshold chosen by the computer that's monitoring this reaction, at which half max fluorescence was met. So we can see that our blue line for gene A reached half max 2 cycles earlier than the red line did. This gives us a delta CT, or the change in the CT value, of 2 for this particular liver cDNA sample. What this tells us is there was more of the blue gene A in that sample than there was of gene B, assuming that the primers are working efficiently in this reaction. Since each cycle of PCR should be, hypothetically speaking, should be doubling that PCR product, then a 2-cycle difference is really a 4 times difference in initial amount. Let's try to break this down. So if gene A was in starting copy number of 1000, okay, in our original liver cDNA sample, then after 1 cycle of PCR, that would double to 2000. After 2 cycles, 4, 3 cycles, 8, and so on, until we reach cycle 6, at which point we're up to 64,000 copies of our particular gene A target. And at that point, we've reached we've reached our threshold fluorescence right here on our graph. We've crossed over this line that the computer has chosen. Now for gene B, we might not necessarily know what the starting copy number was in our liver cDNA sample. We're amplifying off the same template here. But if we look at our graph, we crossed over our threshold mark of what equals 64,000 relative fluorescent units at cycle 8. Okay, so we know that gene B didn't reach the amplification level of 64,000 copies of DNA being made until that cycle 8. What that number tells us, if we walk backwards through these different cycles, is that we had approximately 250 copies back here before cycle 1, which again is about a quarter of how much we had of gene A. So the formula for calculating what the relative expression difference is between these two genes in liver cDNA, which represents the messenger RNAs that are in that particular tissue, is 2 to the delta CT power. In this case, our delta CT, the difference in our CT values between the two genes, is 2. Okay, so 2 to the 2 power equals 4. So that means that gene A in blue here, because it's to the left of gene B in red, is 4 times as abundant in liver cDNA, which represents the mRNA. That means that gene A is expressed at 4 times the level relative of gene B. For scientists and geneticists, more specifically, this is a very useful tool for determining the relative expression levels of genes in single tissues, and as I'm about to show you, between two different tissues. So here we use as a template liver cell cDNA. What happens if we amplify gene A and gene B from, say, brain cell cDNA? If we take a look at this bottom graph, we are using a different template now. We're using brain cDNA, but we're using the exact same primers that we used in the top graph. In this bottom example, our blue line amplifies in much the same way, and that's because gene A I've chosen as a control gene. I actually don't expect it to change in its expression levels in liver cells or brain cells. So a lot of times these would be genes involved in basic cell biology that don't change much in expression from tissue to tissue. They don't have any specialized job, let's say. Whereas gene B is a gene that I'd really like to know, what is it doing in liver cells relative to brain cells? And we can see very quickly that the red line did not amplify slowly as it did up here. It actually amplified pretty quickly relative to the blue line. In fact, its delta or its threshold cycle number is 5, meaning it reached that threshold mark sooner than it did the blue line over here. This generates a delta CT of -1. Now, why was it a positive number up here and a negative number here? Well, in this top calculation, I took the gene B delta, the CT number, and subtracted the gene A number. So down here, we have to do the exact same thing. We take the gene B number, which is 5, and we subtract the gene A number, which is 6. And that gives me a negative delta CT. When comparing gene expression between two different tissues, the way that we're doing right now, between liver and brain, what you sometimes would use is called a delta-delta CT method. That means you're looking at the change in the CT number within a tissue, and then you're looking at the change of that number within two tissues. In this case, if we subtract -1 from 2, 2 minus a -1 is actually 3. So the delta-delta CT in this case is 3. What this is measuring is how this red line over here changes relative to the blue line as we go from liver tissue down here to brain tissue. And you can see the red line jumps from the right side of the blue line to the left side of the blue line. It started out as 2 extra CT cycles here and ended up at -1 relative to the blue line CT cycle down here for a total distance of 3 between the 2 tissues, okay? What does that mean? We have to ask ourselves, is gene B expressed more in brain tissue or in liver tissue? This is sort of strange for a graph where the red line is moving actually down, okay, down the x-axis to the left, where this actually means that there's more of gene A. If it takes less time to reach this threshold mark, that means that there was more of gene B to begin with, right? That it accumulated faster. If we look at the cycle 5 here, at which we met threshold, that must mean that cycle 5 is that 64,000 number that we calculated from the first example here, okay? For that to be true, that means that if we track it back to the starting copy number of gene B in specifically brain cDNA, that because the PCR cycles are doubling each time, that must mean we had about 2,000 copies of gene B starting out in brain tissue. Relative to in liver cells, we only had 250, okay, which is about 8 times less of gene B. And that's exactly what our delta-delta CT is telling us. We use the delta-delta CT with the same formula that we used before for relative expression within one tissue to look at relative expression between 2 different tissues here. So we calculated our delta-delta CT here to be 3. 2 to the 3rd power equals 8. And that means that gene B over here is 8 times more abundant in the brain tissue relative to our liver tissue up here, at least assuming that gene A is a relatively good control, that we know that that's not changing. And so in this way, we can use the 2 to the delta-delta CT method to look at relative gene expression between 2 different tissues using PCR, and we're using these graphs as an output to do it. Knowing now that gene B is 8 times more abundant in the brain relative to the liver means that gene B is probably taking on a greater role in the brain tissue, or maybe that it is actually just upregulated for some particular reason. But it helps us connect gene expression to potential functional differences, which is very useful in biology.